As we know that diabetes the prevalence of diabetes are widely varying worldwide, and by the end of the 2025, the WHO said that the diabetes will be doubled at the time. So it is necessary to take active measures to introduce new drugs officially from better from the natural products. Actually, we have many drugs. Every now and then, pharmaceutical companies come up with a new drug, but actually there are a lot of side effects of these drugs. We are trying to use natural products and use it as to be complementary with with the drugs already in the market in order to lower the side effects of these drugs. At the same time, fenugreek has a lot of properties and a lot of constituents that may lower the blood glucose level. Actually, the traditional medicine use fenugreek for this use, but we want to use fenugreek and introduce it as a new drug or as a complementary method to lower the side effect of the actually presenting anti-diabetic and anti-hyperglycemic drugs. A number of clinical trials showing the efficacy of fenugreek as a hypoglycemic and anti-diabetic agents with limited toxic manifestation. But actually the mechanism of action of fenugreek, we want to know the mechanism of action of fenugreek, and mainly the objectives of this study is the mechanism of action of fenugreek. Does it relies on increased insulin release or decreased glucose absorption from the GIT, or increased insulin sensitivity? This is what we are going to, what I'm going to say in this presentation. Here we use the acute effect of fenugreek administration of the blood glucose level. We have two groups of rats, the non-diabetic group and the diabetic group. For the non-diabetic group, we divide them into 5 groups. We divide them into 5 groups. The untreated one with glypenkylamide, and we use 3 concentrations of fenugreek.
Problem is this cable, which is actually falling off. Is that cable a problem? I think we.
Yes, for the first group, we test the hypoglycemic effect. We test the hypoglycemic effect on the non-diabetic rats, and we divide the rats into 5 groups with different concentrations of fenugreek. For the diabetic group, we introduce diabetes by nicotinamide and streptozotocin to be simulated to type 2 diabetes. And also we divide the group into 5 subgroups with different concentrations of fenugreek and a concentration of glypenkylamide that is similar to therapeutic use in human. For the long-term effect of fenugreek, we divide the diabetic rats into 4 groups. We give the maximum concentration of fenugreek that did the effect in the acute form, and we combine glypenkilamide and fenugreek, but at this time we lowered the dose of glypenkilamide to see, if fenugreek together with glypenkilamide, we can lessen the side effect of glypenkilamide. And we give these drugs for 28 days and measure the blood glucose and serum insulin level. Also, for glucose absorption, we use the inverted intestinal sac method and measure the initial weight of the intestine, the final serosal volume, the initial serosal volume, the serosal fluid transfer in order to see if fenugreek with different concentration affect the blood glucose absorption. Also, we measured the insulin resistance in the diabetic groups. Our results show that when we use glucose concentration by 2.8 millimolar, we found that there is release of insulin. We found there is release of insulin all over the 60 minutes. When we use glucose by 16.7 millimolar, we found there is gradual increase of insulin release all over the 60 minutes. When we use fenugreek in different concentration, 0.1 micromolar, 1 micromolar, and 10 micromolar, we found that fenugreek 0.1 micromolar was similar to the 16.7 millimolar of glucose as regard the insulin release. Similarly, we found this is the percentage reduction, this is the percentage of release of insulin as regard the different concentration of fenugreek. And also with glypenkilamide, and we see that there is gradual increase of insulin release with different concentration of fenugreek and with glypenkilamide. As regard the blood glucose level and body weight, we found that the diabetic group, the blood glucose level decreased with glypenkilamide and with fenugreek, and with glypenkilamide and fenugreek, both together give lower blood glucose level when compared to glypenkilamide alone or fenugreek alone. Effect of fenugreek on rat intestinal glucose absorption, we found that fenugreek 400 milligram per kilogram lowered the glucose absorption when compared to the diabetic group. And when we give the two drugs together, fenugreek and glypenkilamide, we found that there is a much lower glucose level when we compare it to fenugreek alone and when we compare it to glypenkilamide alone. For the biochemical and hematological investigation of the study on the patient with on fenugreek and glypenkilamide, we found that there is no difference there is no difference in the alkaline phosphatase ALT and AST between the groups. Also, when we found the creatinine level, there is no difference between them as regard creatinine level or bond. There is no non-significant difference. Uric acid, bilirubin, and hemoglobin also. For the hematocrit, if there is any difference, this difference was found non-significant between the two groups. For the leukocytic count, there may be slight decrease in the glypenkylamide group compared to the fenugreek group. As regard the basophil, eosinophils, and lymphocytes, monocytes, there is non-significant difference between fenugreek and glypenkylamide when giving to patients in the clinical trial. For platelet count, there is slight decrease with fenugreek, but we repeat it again as we used only 12 patients as a pilot study for this study. When we compare the blood glucose level, actually glypenkylamide 5 milligram give lower range than fenugreek. This means that if we adjust the dose of fenugreek in order to match that in human, or we can give glypenkylamide together with fenugreek in order to lower the level of glypenkylamide and lessen the side effects of glypenkylamide. For fasting insulin level, more or less the same, or there is no significant difference. And also with the HOMA, there is decrease in insulin resistance and may be the same, and also for the hemoglobin A1C. For the lipid profile, actually we have to adjust the dose of fenugreek as we give 1,500 milligram of the capsules of fenugreek that was in the market, and we can adjust the dose in order to give comparable result to glypenkylamide or use it with glypenkylamide to lessen the side effects. In conclusion, there is improvement in the lipid profile after using either the traditional one, the glypenkylamide, or fenugreek, and there is no effect on the renal function or liver function when we use fenugreek for the group of patients for 3 months. Mean that fenugreek has a significant anti-diabetic effect and anti-hyperglycemic effect, and fenugreek can be added to the usual anti-diabetic drugs to decrease the complications and to lower the dose of the drugs and decrease these side effects. What we recommend that we can use the fenugreek in nanoparticles in order to give lower dose and to lessen the side effects, and combine it together with the already present medications for diabetes. Also to make pharmacokinetic studies in order to adjust the dose of fenugreek, either to use the fenugreek as a new drug or to use it in combination with the already present drugs. Okay. acknowledge Prof. Mansour Suleiman, who shared me in this study, and my candidate, Rania Najdi. She did all of the experiments, and also thank for my university and Egypt. Okay, thank you.